Hello, everyone, and welcome to today's Third Thursday webinar. I'm Dave Iverson, contributing editor at the Michael J. Fox Foundation and the moderator for today's webinar, which focuses on the role of gut and the microbiome in Parkinson's disease. As always, you'll be able to submit your questions throughout the hour. There's a little Q&A box that you'll see near the middle of the screen, so just type your questions there, and we will do our best in the last third of our webinar today to answer as many of those questions as we can. And as always, we're also providing the slides from today's webinar for download. You'll see a box called Resource List on your screen, and so you can click there, and then that document will open in a new browser window, and you can save or print them from there. And before we get started, also, just a few tips are on the screen right now, which are some suggestions we have in case we run into any technical difficulties over the course of our discussion today. All right, let's get started with our conversation about the gut and the role of the microbiome in Parkinson's disease. It's a really interesting and emerging field of research that's gotten some attention recently. You may have seen various news articles about this in recent months. And so today we're going to talk about what that means. What is gut bacteria? What is the microbiome? And why might that play a role in Parkinson's disease and in Parkinson's research? And we're going to talk about that connection between bacteria and Parkinson's disease. We'll also spend a little time talking about diet and the role of diet in all of this. And then toward the end, we'll talk about how these new findings may help us to both measure the progression, perhaps, of Parkinson's disease, but also, and perhaps most importantly, could play a role in the treatment of Parkinson's disease as well. Let's meet who's going to be joining us today. Jeff Brunstein is here. Jeff is a professor of neurology and the director of movement disorders at the David Geffen School of Medicine at UCLA. Jeff, welcome.
Thanks. It's a pleasure to be here.
Always a pleasure to have you participate in our event. And joining us as well is Philip Scheper-Jans. Philip is a movement disorder specialist and a neurologist at Helsinki University Hospital. And both Jeff and Philip have been very involved in research in this particular topic area. Philip, welcome to our webinar as well.
Thank you very much for having me. Hello, everybody.
Hello, and thanks for being part of our discussion. All right, let's dig right in and talk a little bit about what we mean when we talk about gut bacteria and this word microbiome. We see a few interesting statistics at the top that there's 3 pounds worth of bacteria in our intestinal tract. So let's first start, if you would, Jeff Brunstein, by just kind of setting the scene for us. Describe what the it is here. What is exactly the microbiome? What does it consist of? And perhaps something about why it's of interest to people like you.
Well, there's the gut is normally filled with, as you stated, trillions of bacteria. And it's a variety of different types of bacteria, and it's a symbiotic relationship with our body, such that it helps us with several functions and digestion, etc. But what's really brought a lot of interest is this connection between the gut and the brain and the communication back and forth. So it's a brand new field that people are just starting to appreciate the potential importance and of what these bacteria do, which bacteria are good and bad, and how that may be affecting brain function.
And Philip, add on to that, if you would, on this last point that Jeff was mentioning, and it's the last bullet point on our slide as well, about the impact on the nervous system. And I think it comes as a surprise to many of us that there are so many neurons in the gut. In fact, as I understand it, outside of the brain, it's where most neurons actually reside in the nervous system that includes the gut. So describe that part of it for us. Why are there neurons in the gut? What role do they play?
Well, I think the traditional way that we thought about it is that the neurons, of course, play a very important role in coordinating actually the movements of the gut when the gut contents are propelled and so on. But they also have a pretty important role, apparently, in sensing the constituents of the food, so they are connected also to the lumen of the gut, and it seems that they are actually, which is something that we learned only quite recently, that they actually very strongly communicate also with the gut bacteria. And so usually we used to think that the nervous cells in the gut were mainly influenced by the brain. So the brain would rather regulate the activity of the gut by sending signals, for example, through the vagal nerve to those enteric nerve cells, and then the gut would like increase or decrease its activity. But the new thing that we have learned now in recent years is that there is actually a very much of information also flowing backwards. So those neurons are sending information back to the brain, and I think we're just only starting to understand about these connections. And so we're now talking about the gut-brain axis, which basically means that there is a strong interaction in both directions between the gut and the brain.
And so, Jeff Bronstein, if it is this two-way, just was going to ask, and you may want to add on to this, Jeff, but if there is this kind of two-way street between the gut and the brain, is that then what places the microbiome of such interest? Because it may have a role in impacting the traffic, I guess, back and forth between the two.
Absolutely.
Yes, so there, yes. No, just go on.
Go ahead, Jeff Bronstein.
Yeah, so absolutely. I mean, there are several ways that this could be influencing the brain and be relevant to Parkinson's disease. One of the things I failed to mention was the effect on the immune system as well. So not only can it communicate, as Philip was saying, directly through these nerves, but they also can release certain chemicals into the bloodstream that can then react in the brain and in other parts of the immune system and the nervous system. And so there are several areas of brain function that people have gotten very interested. The psychiatrists, it seems to affect mood. Cancer specialists are very interested because of the immune system, and now recently in Parkinson's disease. And again, this may be through direct chemicals that are released from these bacteria, direct communication with the nerves in the gut, or releasing of different hormones and other chemicals through the bloodstream that get to the brain.
So given this background and the way in which this interaction begins to play out and affect so many different things, including the immune system, as you were just describing, Jeff Bronstein, let's go ahead then and make this more specific about what we're looking at in the way of the impact of the gut on Parkinson's disease and Parkinson's research in particular. And let's actually start at the bottom of our bullet list here, Philip Shaker-Youngs, if you would, and spend a little bit of time about why this is a natural target in so many ways for interest and research in Parkinson's disease. Starting with the point we see in bullet point number 3 there, that constipation is often one of the earliest symptoms of Parkinson's disease. What is the significance of that, and what intrigues you then in turn about that as a researcher?
So yes, you're right. Constipation is a very important part of Parkinson's disease, and it is specifically interesting because, according to recent research, it is actually frequently present before the onset of motor symptoms. So that is, first of all, one thing that many patients knew for a long time, but that actually neurologists have picked up only again like a few years ago. So drawing their attention to the constipation problem, and it is severely affecting quality of life of the patients. For some patients, it's even the most important symptom, especially when motor symptoms are still rather mild. So, and I think for me as a researcher, the most striking thing about it is that it's such an early symptom, and it may actually, well, I think on average it's about 4 to 5 years that has been reported in the literature, so the delay between onset of constipation and motor symptoms of Parkinson's disease, but it may even be up to 20 years. So the question arises, why is this constipation preceding the Parkinson's symptoms, and since it is now also a recognized risk factor for Parkinson's disease, could it indicate that the pathological process that eventually leads to Parkinson's disease could actually start in the gut? And so it is a very interesting area to look for potential factors that could just kick off the process that eventually leads to Parkinson's disease.
And Jeff Bronstein, is there kind of consensus now amongst you, Philip, your colleagues generally, that Parkinson's does quite likely start in the gut? And is that because of, we know now that there are clumps of alpha-synuclein, the sticky protein that we know kills neurons in the brain, that those clumps also appear in the gut? Is there consensus now that is perhaps the starting point for Parkinson's disease?
Well, I don't think it's universal, but I think there's, I personally believe there's a lot of evidence that at least in the majority of people or in a significant population, there's evidence that not only are there symptoms that start in the gut, I think that's been demonstrated, some very high quality studies, but there's definitely pathology in the gut very early on. Now, whether it's in everybody it starts in the gut, it's not universally I don't think there's complete consensus on that, since there's been some mixtures in some of the articles. But I think there's pretty good evidence that there's early pathology in the gut. There's been synuclein, which is a protein that clumps up that's central to Parkinson's. There's clearly synuclein in the gut. How specific it is to Parkinson's at this point is still a bit speculative, but there's some very interesting studies. So for those of us that study the environment and Parkinson's, the gut is a huge entry point for environmental toxins. So I personally believe that it is a very important place to study.
And does that mean, Jeff, then, that you at least are your hunch, if at this point, if that would be the right way to state that, is that you're looking at those dotted lines, perhaps, that would connect us perhaps from pesticides, the role it might play in triggering the misfolding of alpha-synuclein. And then say something, if you would please, about whether or not it's also your thought that perhaps then it is the microbiome, this gut bacteria that we're talking about today, that enters into that mix and perhaps further exacerbates the development of that clumping process in alpha-synuclein.
Yeah, so that's, I think, the million-dollar question right now is, I think we have pretty good evidence that there are environmental factors that affect the risk of getting Parkinson's and likely the rate of progression. And the question is, how do they do that? One of the ways is through the metabolism of synuclein, and we have some evidence for that. But one very current thought is, and it's very provocative, is that the environment may alter the microbiome, and it's the microbiome itself that is causing this difference, or this clumping of the synuclein through a variety of mechanisms, inflammation, etc. So I think this is where people are very excited about. One, because it's something that we can manipulate and change. So we really do need to understand it. But there are people that have speculated, for example, people that smoke cigarettes have a much lower risk of getting Parkinson's disease, and there's some evidence that smoking may alter the microbiome, and maybe that's how you lower the risk of getting Parkinson's disease. On the other hand, it may be the other way around, is that having Parkinson's disease may alter the microbiome, and we're really not sure which is the chicken and which is the egg at this point, and that's where a lot of research needs to be done.
And we'll continue that chicken and egg question about whether or not affecting the microbiome might in turn affect the progression of Parkinson's disease, or whether it's the other way around as we progress throughout our webinar today. But before we explore those in somewhat more detail, Philip Shaper-Jans, if you would also touch on this last point on the slide before we move on, that also is significant within Parkinson's disease, and why the gut is so important, because we know that our stomach empties more slowly in Parkinson's, And that has a big effect on how well we can treat the disease because of medication absorption. So that's yet another reason, it sounds like, why this is such an important area of research.
Yeah, so I think actually this is probably one of the most important things to remember when treating patients with advanced Parkinson's disease, that independent of the question what the role of the microbiome might be in this context, so we see that even in early disease, about 60% of patients show slow emptying of the stomach, and this fraction increases as the disease increases. So what that basically means is that the frequency with which we or the patient takes in, for example, levodopa tablets, that this does not necessarily that's not necessarily the same frequency as it happens in the gut. So the problem is that levodopa is not being absorbed so much in the stomach, but rather in the small bowel. So what we need is that the tablet will be moving forward from the stomach into the small bowel. So that can be impaired in Parkinson's disease, and so it can happen that, for example, tablets just don't reach the small bowel in due time, and so people don't experience the benefit from levodopa, or they experience it in a delayed way. Or we can even have accumulation of tablets in the stomach, and then those are being basically released, let's say, in an uncontrolled fashion, so all at once, which can then lead to uncontrolled dyskinesia. So it seems that actually this function of the upper gastrointestinal tract, so the esophagus and the stomach, is a very relevant factor when we are treating patients with motor fluctuations.
And Philip, does that also mean that this is another active area of potential research, whether or not the microbiome, the bacteria in the gut, are affecting that absorption process?
Well, definitely it is something that needs to be further studied. So I'm not aware currently about any study that has analyzed the whole microbiome, let's say, of the stomach in relation to these symptoms and problems. But, for example, there is some indication that Helicobacter pylori may play a role and may be connected to this kind of dysfunction in the stomach and to the disturbance of the resorption of medications and the, let's say, problems related to motor fluctuations and lack of motor benefit from levodopa.
That's yet another bacteria that appears in the gut. All right, let's dig deeper now into this connection between Parkinson's and the gut. And Philip, pick up on the first point, that people with Parkinson's may have less of a bacteria that helps protect against the environmental toxins that Jeff Runstein was describing a moment ago. This has been an area of research for you, that there may be lower levels of a particular bacteria called Prevotella, I think. And tell us something about what you've learned about that and whether or not having less of that bacteria and how that may tie to an inability perhaps to protect against environmental toxins.
Sure. So maybe I just give a quick introduction in how we actually analyze the microbiome. So what we do is that we read out a certain segment of the genome of the bacteria that are in the gut, and we do that several thousand times per sample, and then we basically compare these sequences to a database. So we then have numbers that represent the abundance of different types of bacteria in our sample. So we can have like a picture of who is there and what are the numbers and who's there, like what bacteria is a lot and what kind of bacteria is very rare. And so then we can compare that between patients and controls. And that's basically what we did in our first study. So we looked at Parkinson's patients and we looked at controls and compared the composition of their microbiome from feces, and the main finding that we had was that this group of bacteria called Prevotella, so like a family of bacteria, this group was reduced by almost 80% in Parkinson's disease subjects. So unfortunately, we were not able to do an analysis of the functions that these bacteria had specifically in our patients and controls. But we know from the literature that this bacterium, for example, feeds on the mucus that is like a mucus layer that is on the inner surface of the gut. So, and this mucus is considered to be protective. To protect us from toxins and so on, and so prevents them from entering into the tissue of the gut and from entering the bloodstream. And so there is some evidence that when this mucus layer becomes thinner or destroyed, that this may predispose to an inflammatory reaction in the gut mucosa. And so one hypothesis is that this Prevotella reduction may reflect changes and dysfunction in the mucus layer of the gut, and so this could be theoretically related to higher exposure to environmental toxins, for example. And in other studies, for example, from the United States or from Germany, also other bacteria were found to be differentially abundant in Parkinson's disease and control subjects. And in those studies, bacteria that were considered to protect from inflammation were found to be reduced in Parkinson's disease subjects. So there is some agreement, let's say, around the axis of gut mucus protection and inflammation in this respect, although there are also significant differences between the studies that have looked at this subject.
And Jeff Brunstein, again, this gets into this sort of chicken and egg question, because I suppose you could look at findings like this and you could say, well, is it having less of that bacteria that makes you more susceptible to environmental toxins and their impact, or is it the environmental toxins that lessen the bacteria? It could go either way, right?
Absolutely. And furthermore, we know that at least in some of the studies that people that are just constipated and don't have Parkinson's have a different type of bacteria in their gut, which has some similarities to some of the studies that have been found in Parkinson's. So is it possible that Parkinson's disease is causing the constipation, which is changing the microbiome secondarily? So that's one possibility, but the intriguing possibility is the reverse. And there's a recent study from the Caltech group, which I'm sure we'll talk about in a minute, which suggests that changing the microbiome specifically can alter the disease process. And I think whether that and how that is changed, how that microbiome is changed, is it through the environment, likely diet, etc. So which is cause and effect is still not clear, but it's so provocative of a mechanism that's why I think it's gaining so much attention in the research community.
Well, let me just mention very briefly the study you referenced, and then Jeff, you can amplify from that. But as I've read and understood this study, at least, what they did at Caltech was they had some laboratory animals which were bred to have too much alpha-synuclein, the culprit, the sticky protein involved in Parkinson's disease. But the two sets of mice were different in that one had a normal microbiome, normal gut bacteria, and the other group of mice didn't have a normal microbiome. They were bred to be essentially germ-free, sterile microbiomes. And then they found that in these two sets of mice, the mice that didn't have a microbiome at all could still function quite well on various motor tests that they devised for this lab experiment, whereas the animals that had a normal microbiome didn't do as well. Even though they both had the same levels of alpha-synuclein, it was the ones that had the gut microbiome that didn't function as well. And that's what you're suggesting, Jeff? That's what's so intriguing, is it may be the gut that triggers the problem with alpha-synuclein.
Right. Well, so in this model, this is a model that's been very heavily studied here by my colleagues at UCLA that work with the Caltech group. So we know this model well, which they're genetically modified to make too much human alpha-synuclein. And what this study has shown is that the is exactly what you said, is if you've had no microbiome, they wipe it out, the pathology in the brain and the motor effects were much less than if they had their normal microbiome. They also found that if you take the microbiome from Parkinson's patients and transplant it in, they had more of a pathological problems and motor problems than if you gave the microbiome from healthy people that didn't have Parkinson's, at least in most of the groups, not in all of them. And again, they're suggesting through some additional studies that how this is working is that the bad microbiome is making some chemicals, some metabolites called small-chain fatty acids that are causing inflammation and altering the pathology in the brain. This is, again, very intriguing, although it contradicts some of other data that have been shown by others of the effects of small-chain fatty acids, and maybe I'll let Philip address that a little bit more in-depthly.
Well, before
so
you do that, actually,
sorry,
I was just going to suggest before we get too much into too much detail about that, because already we're getting lots of questions from people, not surprisingly, about, well, what can we do then? And how can we alter our microbiome? And I want to move to that in just a moment. But go ahead, if you would, Philip, just mention briefly that this is, again, one of these intriguing things, because sometimes those short-chain fatty acids that Jeff was mentioning have been shown to have a positive effect on the immune system. So again, lots to figure out, intriguing, but lots that still has to be sorted out.
Yes, correct. So I think it's very complex, and when we look at human studies, the reports that we have so far point a bit in a different direction than the animal data from Caltech, but there are several different types of short-chain fatty acids, and they may have different effects, and it may also be relevant in which relative concentrations those are applied. So we need to learn more about it, but the data that we have showed that in Parkinson's patients, the short-chain fatty acid level seems to be decreased, which is in contradiction to the mouse study that we received from Caltech. But it's only very preliminary data, and it needs to be replicated, obviously.
And Philip, pick up our last point, and then we'll turn to this question of what people might be able to consider doing in terms of diet to alter their microbiome. But we see this other point here on the screen that we should address, which is that other kinds of bacteria, and this is where it gets tricky. There are thousands, if not tens of thousands, if not millions of bacteria that may be involved here, and yet another one there seems to be a connection between the levels of that bacteria and a particular symptom such as gait and balance difficulty. What do we know about that?
Well, yeah, so when we did our study, we decided beforehand that we wanted to check whether we could find any connection between microbiota and the actual clinical symptoms of the patient. So, and we have known that, I mean, everyone knows that Parkinson's disease is a very heterogenic disease. So different symptoms may be expressed very differently in different patients. So, for example, we have patients that have the main symptom of tremor and may otherwise be pretty healthy and may be able to walk almost normally and don't have so much stiffness and so on, whereas we see other patients that maybe don't have tremor at all and instead suffer from rigidity and early problems with balance and gait difficulty. So those are actually named, for example, those extreme cases like a tremor-dominant case patients and a postural instability and gait difficulty phenotype, where although those phenotypes have been discussed and they're not necessarily the only ones, but those have at least been mentioned in the literature. And so we contrasted those in our study, and what we found was that a family of bacteria called Enterobacteriaceae, which contains several, like many bacteria, including those that are thought to be pro-inflammatory, some bacteria of this group, those were increased in patients that expressed those gait difficulty symptoms. So, which is interesting because we also know that, for example, those patients having predominantly gait difficulty and rigidity and stiffness, they tend to have a bit worse prognosis and to progress faster. They have a higher risk of, for example, getting cognitive impairment and dementia. So there is maybe a different course of the disease, and our data suggested that this may also be reflected in the microbiota. But as previously, we don't know what is the chicken and what is the egg in this case. But I think it's an intriguing finding that to find a connection between the clinical motor symptoms of subjects and gut microbiota.
All right, that leads us nicely into this question that I think everyone on our webinar today would like to hear more about, which is there anything we can do about that as people living with Parkinson's? And as we've already indicated, it's tricky because there are so many different kinds of bacteria, and some may be okay, and some may have an adverse effect on Parkinson's. We don't know yet. It's hard to know to sort them out, which we might want to promote, which we might want to diminish. So it's a complex area, but understandably, everyone would like to know, well, is there something I can do to have a more healthy microbiome for health reasons generally, but for Parkinson's specifically? And as our Director of Research Communications, and Maggie Kuehl, has pointed out to me in our window here on the side, it says, so many questions on probiotics. So everyone would like to know, Jeff Ronstein, whether or not they can or should do something to boost probiotics, things like yogurt or other things that would affect the microbiome. What's your put on your clinician's hat now as someone who sees patients every day? What do you say to people about what they can do or should perhaps do to change their microbiome?
Well, we're still, as the whole discussion, there's a lot of discussion about which bacteria we want and that sort of thing. So I think the absolute answers is not known. But I think we do know a few things that are important. So the first thing is even if you take a lot of probiotics, it doesn't necessarily mean you will maintain those bacteria that you've ingested. And so there's been a big movement to what's been called prebiotics, which are basically the food that you're feeding the bacteria. So we do know that things like high soluble fiber that's in a lot of fresh fruits and vegetables, in a very plant-based diet, low simple sugars is a good thing, and unsaturated fats, not the saturated, you don't want the meat fats. We know all those things seem to promote what we think are bad bacteria. So probably what's more important than probiotics is the concept of a very healthy diet, the high fiber, the low meat fats, things like that are extremely important in maintaining a healthy microbiome. So and we do know that a healthy diet is important for your general health, not just for Parkinson's disease. So I think we can safely say, a very high diet, some people would even say a more of a plant-based diet, less in red meat, is going to be good for you no matter what. And whether it's through the microbiome or not, we're still not sure. I think taking probiotics at this point by itself, I think we just don't know unless we're in certain situations. When you've taken antibiotics, there's some rationale for taking it. When you're taking antibiotics, and then there's some bad bacteria like C. diff and things like that, where changing the microbiome, we have a better idea. So I think at this point, a healthy diet is number one, and number two, and we'll have to wait to see, whether taking probiotics by itself is enough to make a difference.
And Philip, what would you add to that as far as the particular problems that people in Parkinson's tend to have in terms of constipation? Or is there anything that could also be done as far as that stomach emptying problem that you mentioned earlier? Are there dietary guidelines that could help with either of those issues that are so prevalent in people with Parkinson's?
Well, I think it is commonly agreed upon that high fiber diet, of course, in combination with also enough fluid intake is beneficial for the symptomatic treatment of constipation. So that is definitely something that also from this standpoint is suggestible for Parkinson's disease patients. In terms of, for example, gastroparesis, what I usually recommend to my patients is also to reduce the amount of fat in their diet, because it is known that this slows down gastric emptying. I usually suggest to eat smaller portions, but many times a day. And there is some evidence that also foods that decrease the pH level, so that increase the acidity of the gut, could also aggravate the gastroparesis. So, for example, reduction of citrus fruits, and so it could be beneficial. But those are like symptomatic suggestions that I frequently give to my patients. There is one study recently that specifically investigated the potential of a probiotic combined with a prebiotic in the treatment of constipation associated with Parkinson's disease. It was a placebo-controlled study, and which had a clearly positive result. So patients' constipation was improved. It's in a study from Italy. The unfortunate thing is that the combination that was used in this study is not commercially available. So far, I think there is no really commercially available probiotic or prebiotic that has been studied in Parkinson's disease patients. So usually I do not specifically recommend any probiotics for my patients.
Jeff Brunstein, a couple of other dietary questions, and then we'll move on to an additional topic. But one person asked about the impact of dairy on the microbiome in particular, but on Parkinson's in general. Any comments you want to offer in that area? And also perhaps if you would touch on the question of eating organically because of your particular interest in pesticides. I'm curious if you have any recommendations that you give to patients in that regard too.
Yeah, so there's not too much research on the effect of dairy. There was a very nice study out of Hawaii. Webb Ross did a nice study, and he demonstrated that people that drank a lot of milk actually had a higher risk of getting Parkinson's disease. And there's been a lot of speculation of why that is. And one of them is that pesticides are found in milk and other toxins, and maybe this is a route for getting increased toxins such as pesticides. And there's, in my mind, there's little doubt that a very high exposure to pesticides, especially in farming communities, et cetera, way above and beyond what the average person gets exposed to, can markedly increase risk, at least in certain pesticides. So I think you definitely want to stay away from high exposure areas and applicating it, et cetera, applying it. And I'm not a black and white person, and what I tell people is what I do for my family, which is to eat organic as much as you can, which is reasonable, especially certain fruits and vegetables, which traditionally have a high amount of pesticides along with it, residues called the dirty dozen. And so try to avoid that. But, I think we don't know if very low levels how much of an effect that has. And again, with relationship to this conversation, we're still not sure how much of an effect the pesticides have on the microbiome. As you know, there's hundreds and hundreds of different pesticides, and they're not all the same. So they're going to have different effects as well. So in general, I try to minimize exposure to pesticides, eat as much organic, which is reasonably possible, and other than that, the one last effect I would say about dairy is there is protein in it, and protein does interfere with the absorption of levodopa. And there's a classic study where they measured levodopa absorption with milk protein versus water, and there was a dramatic reduction in absorption of levodopa. So probably a good idea not to take too much dairy at least when you're taking your medications.
And one last question on this general topic, and then we'll move to our last topic area and take more of your questions in just a moment. But I know you've done some research on this, Philip, so let me give this one to you. It's a question from Ruth, who asks about the benefits to reducing the H. pylori bacteria through antibiotic treatment in Parkinson's. That's something that I think, actually, Jeff, I know you've looked into that too. So this is a particular bacteria, and do we know that reducing it is important in terms of how well people are able to do when they have Parkinson's disease, or how well their medications actually function? Philip?
Yeah. So just to clarify, I didn't do that research myself, but I read about it. So H. pylori is a bacterium that has been found in the stomach and is related to inflammation of the stomach, so what we call gastritis. And so it has been associated several times also with an increased risk of Parkinson's disease, but not all studies were able to confirm that. But it seems that it is definitely related in Parkinson's patients to the severity of motor symptoms. So when we look at different Parkinson's patients and compare, for example, patients that have Helicobacter with patients that do not have Helicobacter, then those with Helicobacter tend to be in a worse motor state, more progressed, more and worse medication response, more off time, for example, and worse fluctuations usually. And this has been attributed to the inflammatory process that this bacterium may induce, and it is thought that it may impair the resorption of levodopa, and it could also, through the inflammation, decrease the peristaltic, so the movement of the stomach, and thus aggravate the gastroparesis. And there are also several rather small but also placebo-controlled studies where it was compared how eradication of Helicobacter using broad-spectrum antibiotics affects those symptoms. And there seems to be an indication that when you eradicate that bacterium, then you have an increase in on-time, or you can have an increase in on-time, and you can have a reduction of those events that we call delayed on, so where the response to medication comes on late or doesn't kick in at all. So these symptoms may be reduced when you eradicate the Helicobacter. But of course, some people argue that when you do these kind of antibiotic treatment, you don't only eradicate Helicobacter, but you also, of course, change the microbiome more generally. So, and this is not really clear whether this is a specific effect related to the removal of Helicobacter or a more thorough change of the whole microbiome. So we don't really know that.
All right, thank you. Let's explore kind of our last topic area, and we'll continue to take your questions, of course, as we finish our last 15-20 minutes or so of this. But this is a key area that we want to make sure we investigate as well, and that's how these various findings will impact our greater understanding of Parkinson's, but also our treatment options. And one of the things that's so intriguing, I think, about this, Jeff Bronstein, is that in our overall goal of being able to slow the progression of Parkinson's disease, the gut is an easier target in some ways than the brain, right? Because we can get drugs to it right away, as opposed to having to travel through the gut and up to the brain and across the blood-brain barrier and all the rest. So there's a lot that's appealing. If indeed Parkinson's starts in the gut, we can get in there in a more ready way, it would seem like. So from a researcher standpoint, again, say something about how it represents such an interesting and intriguing target.
Yeah, so it's very complex, and I think we're right at the very beginning of these studies. So it's premature at this point to say anything other than there are intriguing possibilities. So you can imagine that if the gut microbiome does alter, the risk of getting the disease or the progression of the disease, this is something we can manipulate. We can manipulate with diet, we can manipulate with antibiotics, and we can manipulate with probiotics. So first we have to determine what are the best bacteria. And you have to think not only of what it is for Parkinson's, but for other things like heart disease and autoimmune disease and cancer, because we also don't want to set up bacteria that's good for one system and not for the other. So I think there's a lot of work that needs to be done before we have unintended consequences. And it's also important to understand how we're going to figure out what really causes Parkinson's. And so we need to know those mechanisms. And, it's playing another piece in the puzzle. So I think all these things are really important in understanding the causes and how we may treat it. The other part that I think is important as well is when we do clinical trials. So even if we do a clinical trial on something that has related, not at all related to the microbiome, say we're doing an immunotherapy, it may be important to know what microbiome people have, because you want to have as homogeneous as a patient population, and maybe at some point we even want to match people in the studies that have the same microbiome, so that if we give treatments and everything, we're more likely to see an effect of that treatment, and it's not going to be obscured by differences in microbiome. So there's so many ways that this may be playing a role. It's going to be essential to have a better understanding of this whole process.
And does that mean, Philip, then, that the microbiome, the bacteria you have in your gut, may be useful in addition as a potential target, but as Jeff suggests, it may also be a way of dividing people up so that we study them differently, and we know, that we're testing a drug with people who have a particular kind of bacteria versus another, and that it may also be a way to, is it also a way that you could perhaps further be able to diagnose Parkinson's or different types of Parkinson's, a way to be a kind of biomarker that can allow us better diagnostics, better tracking of disease progression? Are all those things possible?
Well, I think the most important thing that we need to figure out is whether we could use an assessment of gut microbiome composition, or let's say function, to diagnose the early stages of Parkinson's disease before people develop motor symptoms. So that could, for example, like hypothetically mean that if a person suffers from considerable constipation and there is no other explanation, we know from epidemiological studies that this person has about a 2 to 4 times higher risk to get Parkinson's disease as compared to a person that does not have constipation. So, but we're not able currently to figure out individually whether this individual will get Parkinson's disease or not. So, and if we had some means to derive that information, for example, from a fecal sample, and by analyzing the microbiome, that would be a huge step forward, because it would basically allow us to identify those patients very early, and to also, for example, select them for clinical trials and to try to slow down the course of Parkinson's disease. So that's the eventual goal, I think, that we have. So in other terms, I completely agree with Jeff that it would be very important to achieve more homogeneous study groups for clinical trials, because I think that is maybe one of the reasons why many clinical trials in Parkinson's failed so far, is that people with very different phenotypes, as a different expression of symptoms, that may actually represent different pathological processes as well, maybe underlying the disease, that they have been clumped together in the same group, and so we might not be able to pick up any disease-modifying effects when we put all patients together.
In other words, a drug might well work with a certain group of people with Parkinson's who might have a different microbiome than another group. So it's another way in which we often talk these days about precision medicine. It sounds like you're both suggesting that the microbiome may be another way of differentiating how to be precise with how we test drugs, and in the end, what drugs we give people. Is that right?
Definitely.
Absolutely.
All right. Thank you both. Let's move to just some general questions and answers that we can provide to people that have come up over the course of our hour, and we'll return to any more questions about probiotics or anything else that people may have. Jeff, a question about whether or not there is a connection then between Parkinson's disease and other kinds of gastrointestinal problems. If the gut is important to Parkinson's, does that mean that there is a connection between having Parkinson's and perhaps having other diseases that affect the gut, such as Crohn's disease, irritable bowel syndrome, celiac? Is there any kind of connection between those various conditions and Parkinson's?
So far, I'm not aware of any connections that's been made with any of those other diseases. So far, the only connections with the gut that I'm aware of are, the constipation and H. pylori, which we've already discussed. So I don't think there's a strong connection that has been found yet.
And Jeff, another dietary question about fermented foods. So you've gone through yogurt, organics, and dairy, but what about sauerkraut?
I don't know. Maybe Philip can answer that question.
Okay, Philip?
Thanks, Jeff. I'm afraid I cannot tell you anything about this. So I've not read any study that has dealt with this question.
But you may want to take breath mints afterwards.
The sauerkraut question has stumped the panel. A number of people are bringing up two areas that we should give a little more attention to. One is this question of microbiome transplants or fecal transplants. This may, Jeff Bronstein, be connected to the Caltech study that we referenced earlier, which is part of that study, and I think you referenced this. They took microbiome samples, fecal samples from people with Parkinson's, placed them in laboratory animals, and those animals then did worse, which again is intriguing and suggests the role that the microbiome may play. I'm not sure if that's exactly why people are raising the question of microbiome transplants, but respond if you would, if there's anything else you can add about microbiome transplants or that you think is more relevant in terms of the Caltech study.
Yeah, so there's actually a fair amount of work being done on fecal transplants, and that's because there's a disorder called C. diff or C. difficile, which is a bad bacteria that people can get overgrown when they have taken antibiotics multiple times. One of the reasons why you don't want to take antibiotics all the time if you don't need to, because you can get this overgrowth of C. difficile, which can cause diarrhea and a lot of GI problems. And often antibiotics don't work for those, and fecal transplants, taking people normal microbiome and transplanting it into a patient with C. diff, can cure it. So there's been a few observations made outside of Parkinson's. For example, fecal transplants from a slender person to an obese person has actually been reported. And again, it's very anecdotal, but they've lost some weight. And so that maybe the microbiome has effects in other areas such as weight loss. There's been anecdotes with other disorders as well. So the Caltech group, which when they transplanted Parkinson's microbiome to the mice, they seem to do worse, and when they transplanted healthy controls, does lead to that concept of, should we be doing this in people? And the answer is we just don't know yet. Mice are very, very different than people, their immune systems, etc. So I think it's a little premature to start doing that, but it does have that possibility. But again, once you transplant, if you do not have a healthy diet, you're not going to maintain that healthy microbiome. So you can't just transplant. You can't just take probiotics. You need to change and have a healthy diet and lifestyle to maintain a healthy microbiome.
Great point. Philip, anything you want to add on that subject and on the prospects of being able to do microbiome transplants?
Yes, so I think that microbiome transplants is a very interesting field that needs further study, but I would also ask for caution with this because I know that there are commercial agents that are providing fecal transplants also to Parkinson's patients. And so there is absolutely no evidence currently that this is beneficial for Parkinson's disease. And it must also be considered that although the C. diff, so the Clostridium difficile treatment with fecal transplant is very effective, it must be considered that the microbiome that those patients have before the transplant is extremely abnormal. So it has undergone a huge change that, and these changes are much stronger than what we have found in Parkinson's disease patients. So I think when at some point we will go to study fecal transplant in Parkinson's, we first need to learn more about the specific changes that we see in Parkinson's and to then better select, let's say, a potential donor or a certain microbiome that we want to transplant. So, and this is not currently done for the standard fecal transplants that are applied. So those are not specifically for a certain disease, but they're rather a generic transfer of a supposedly healthy microbiome. And we don't actually know very well what is being transplanted with these microbiota. So we don't really know whether this microbiome could actually predispose a person to some other disease years ahead. So we have no data about it, and I strongly discourage people from getting fecal transplants for PD outside of clinical trials.
So I just want to add to that.
Yeah.
Yeah.
Go ahead, Jeff.
So I hadn't heard that there are places already offering this, but it doesn't surprise me. And I want to just echo exactly what Philip says. I would not recommend it because we don't know what harm it might do. And I will say that anecdotally, I've had 2 people in recent years get fecal transplants for C. diff that have Parkinson's disease, and it had absolutely no effect on their Parkinson's disease, one way or the other. So it's definitely not the magic cure for everybody, I will say that. And so I would strongly recommend not doing this as well because you don't know what harm you may do at this point.
I really appreciate both of you sounding that cautionary note. I think it's something we come across in Parkinson's disease often, where people are offering surefire breakthrough cures online that are recruiting people for those cures, often expensive ones. And I think whenever you see those advertised, buyer beware is the appropriate response. So thank you both, Philip and Jeff, for underlining that very important point. We have to begin to wrap up our conversation, but I want to ask you both just for a closing thought, if you would, a brief one. But I think it's often, as intriguing as this is, it can often be frustrating for people who hear this because you do want to be able to do something, perhaps not a microbiome transplant, but you'd like to be able to have a takeaway. You know, what can I do? Whether that's changing your diet or altering lifestyle in some way or taking medication in a different way. So let's leave people with a thought about what we can take away from this, either in terms of what people can do in their daily lives, but also perhaps pointing to intriguing research that if the answer isn't there today, we hope it will be someday in the tomorrows of the future. So Philip, just a last closing thought that you'd like to leave with people today.
Well, yes, I think as a take-home message, although some degree of patience is necessary before we will see any, I think, clinical applications of those microbiome studies, I think what people should take home with them is that by using a healthy diet, consuming a healthy diet, they can be rather sure that they will not only benefit their general health, but this would also probably support and their improve their Parkinson's disease symptoms, at least in terms of gastrointestinal symptoms, and possibly also medication response. So most likely also their Parkinson's would benefit from such a good healthy diet. And in terms of research, I mean, I'm just extremely excited that I have the chance to be part of this exciting endeavor, to really be able to, from actually zero, start to explore this field in relation to Parkinson's disease, which is so different from all other approaches that have previously been followed to tackle the mystery of Parkinson's. And so I promise that at least here in Helsinki we will do our best to learn more about it, and we always have in mind the final goal of getting Parkinson's disease out of the list of unexplained and basically unstoppable diseases.
Well put, Philip. Thank you. Jeff, last thought?
Yeah, so I echo what Philip just said, especially about healthy diet, et cetera. I think it does emphasize how complex the causes of Parkinson's, not just the pathology and the symptoms, and there's probably not going to be one answer for everybody, but there's going to be many small answers for the ultimate cure for Parkinson's disease. So I think there's a couple things that people can take home and do. One is we know a healthy diet, what we've talked about already, can not only help your symptoms now, but potentially help the underlying disease process. And the other aspect you can do is if you're open to participating in research, you should do that because that's how we're going to find these answers. So I don't want you to go I don't emphasize people to go to one of these shops that's marketing the cure, but I am emphasizing that if there's a legitimate study going on and you're able to participate in it, that's going to help bring us the answers. So we have to do this research in people eventually because the animal research doesn't always translate one-to-one. So that would be the only addition I would have is if you're able and interested in participating in research, definitely get involved.
Thank you, Jeff. And one way people can participate in clinical research or to find a clinical research trial near them is to use our foxtrialfinder.org system, which allows you to find trials in your area and to match them up with your particular interests. I want to thank everyone for their participation today, all of our online participants, and particularly, of course, Jeff Bronstein and Philip Schaefer-Youngs for their participation. Jeff and Philip, thanks so much. Thanks not only for participating today, but of course for the work that you are doing. We'll be sending a link to the webinar on demand so that you can listen again or share as you'd like. I will also mention that we've mentioned that Caltech study a time or two in this webinar. We have a podcast specifically about that study that you can find on our podcast page at the Michael J. Fox Foundation website. So if you're particularly interested in that, you can also reference that. And we'd also like to ask you to mark your calendars for our next webinar, which is on February the 16th, where we'll talk more about genetics, what we know right now about Parkinson's heritability and how the genetics revolution is transforming our approach to Parkinson's disease. Thanks again all for participating. Until next time, I'm Dave Iverson.